Welcome back to SBT 458, Principles of Industrial Biochemistry. Today we're looking at kinetics of microbial growth and product formation in biocatalysis. The things we are going to look at today actually are things which we have already seen in general biochemistry, looking at those fundamentals of enzymology that we did, except that today we are going to look at them with the eye of an engineer, may say a biotechnology engineer, so that some of those things which you saw, we are going to rather think of them more specifically as being applied in industrial engineering. And so our lecture is going to be a bit mathematical, but very brief because you know how mathematical things are. They are straight with figures, formulas, and then you get to understand how to apply them, and then that's it. So we will by the end of the lecture, get some practical applications of those things that we did with some enzyme kinetics, how those things are used in the engineering of biotechnology systems. Say engineering of say bioreactors, which are used in the industries for say fermentation processes, or different production processes in industries. And so we'll first take a look at say kinetics of microbial growth, which we have seen even multiple occasions in this course. I think we have seen it maybe about maybe twice already, and you have seen it multiple times already in microbiology. You keep referring to that kinetics of microbial growth over and over again, and it is that important, and is very much applied in industry. Now, the first thing we'll look at is what we refer to as the Mannord equation, which is very much like the Michaelis-Menten equation. You remember we did look at enzyme kinetics, and we refer to this kinetic curve, and we refer to say the Michaelis-Menten constant, which is the concentration of a substrate at which half of the maximum of the maximum rate of reaction is reached. So you have just the application of that Michaelis-Menten kinetics, but this time around by Mannord, which is being referred to as the Mannord equation, where you have a specific growth of, the specific growth rate of a microorganism being expressed in relation to substrate concentration. So that instead of us referring to that constant, that concentration of substrate at which half of the maximum rate of the reaction is reached as a Michaelis-Menten constant, this time around is being referred to as the half saturation constant, because it's not Michaelis-Menten equation. And so but if you think of it, it's just the same thing, but it's because it's being applied in a different context. And so that is why you are just calling it a half saturation constant. But if you think of it, actually, it's a Michaelis-Menten constant, which is just used, practically applied in a different way. Now, so of course, then you have that mu max there being referred to, say, the maximum specific growth rate of the microorganism. So this time around, it's not the maximum rate of reaction, but the maximum growth rate. So as we continue, you see when we are looking at that particular the growth kinetic curve of the microbes, then you can relate to that to say that yes, there's a maximum growth rate that a microorganism reaches in that curve, and of course you have on that horizontal axis the substrate concentration. That's the concentration of the nutrient which is introduced into the bioreactor. It is on that nutrient that you have the microorganism feeding and making use of that nutrient to produce, particular product of interest that can be harvested. So you see that we've started looking at it mathematically, and in terms of engineering, you are still using that knowledge which you have gained already in microbiology and general biochemistry. Now, yes, the bacterial growth curve again, and this time around you look at it as you have a lag phase, which you understand. That is a stage where you have little or no cell division occurring. At this stage, you have synthesis of some new enzymes because the microbes are sort of preparing to start growing up. And then you have that exponential phase. That is where the growth occurs, the growth of the microorganisms multiplying in numbers. And then you have at a stage where you refer to as a stationary phase. So you see that is where you reach the maximum growth rate that we refer to, the mu max. At this stage, you do not have the microbe cells growing, but what you do have is that you have the number of cells, sort of almost double, and at regular intervals, but the growth is stationary at this point. And then you have the number of live cells remaining constant over this stationary period of time until you get to a point where you're having some opposite of what was occurring with the growth or the exponential phase. You have that happening with the last phase, which is the death phase, primarily because of depletion of nutrients in a bioreactor, and also because you do have these cells which have doubled over this period of the exponential phase, where they've doubled in their numbers and increased, and then when they get to the stationary phase and they keep undergoing their metabolic processes, they produce some toxic metabolites as well. And so toxic conditions are then created in this phase, and so the cells begin to die very fast. That's a death phase. Now, we did refer to this several times, that you need to know this growth curve, the growth kinetics of a particular bacteria you are using, in order for you to be able to optimize your system, know when the time to set for your product to form, and the time you are to stop the system and harvest, particularly if it is with a batch system, because we looked at these different types of systems of production. Now, mathematically, the batch system can be expressed in terms of cell concentration. And so if you take N as a cell concentration at a particular time, and you have N0, which is the initial cell concentration, which is like if you have say a starter culture, and then with this starter culture you then introduce it. After it grows over a period, then you introduce it into the bioreactor. you want to know at a particular time how much of, what will be the concentration of say, the bacterial cells or the microbial cells at a particular moment in time. You can express this with such a mathematical equation where you have all the terms being explained here, where you have the mu being the specific growth rate at that particular time. The T is the time. The N0 is the initial cell concentration. That's starter culture, how much of the cell is in there, and then you have the cell concentration at that particular time that you are referring to. So it means that with this mathematical equation, you can actually determine at every point in time in the production process how much of microbial cells are there. How much, what's the concentration of microbial cells maybe after 30 minutes, after 1 hour, 30 minutes, you'll be able to determine that, expressing it mathematically this way. Of course, this is particularly important for the batch culture because you remember that in the batch, everything is fed into the system, closed, you wait until the last end, everything is done. The system stops before you start to do any harvesting so that you should be able to predict when the process should be stopped, when you should start harvesting. So you do not have, say, that the last end, which is the cell death, getting so much deep into the process and rather contaminating your product, because then you start to have toxic metabolites that are being produced by these microorganisms together with the cells that are dying, and toxic conditions are then created in there. Now, if you then look at it in terms of the continuous culture, we also did look at, the continuous system where you had the flow of nutrients into the system continuously. That was what we looked at as the major modern system, which is used to increase yield. Then you want to think of, in that process of continuous flow of medium, that is, the feeding of the system, that continuous flow, you want to think about what effect, the dilution effect that can have on the system. And so we refer to what we call the dilution rate, which is the rate at which fresh medium is added to the culture. And it's a ratio of the flow rate. To say that maybe you are flowing, the medium is flowing at, say, 100 microliters per second, for instance. That is actually fast if you're looking at production over hours. That is a very fast flow rate to say. And you have that flow rate of the medium, the ratio of it to the volume of the culture. So then what it means is that dilution rate will control the rate of the the growth rate of the microorganisms. In terms of the fact that, see, the medium is the feed, is the nutrient on which the microorganisms are feeding before they are able to, produce, the product. So the amount of the feed that comes into the system over time will determine the growth rate of these microorganisms. So you see, all of this is considered when developing, such a bioreactor, and the pump to it, the pump which will be pumping the feed, the medium, into this bioreactor. At what speed should it be set if you are setting up the system, at what speed should it be set. And you actually will you have to do some calculation of the amount, the volume of the culture which you have, which the volume of the medium which you are actually connecting to the pump, and at the same time the volume of the culture, I mean, that is inside of the bioreactor, because then you have to get an amount of a medium that can flow, and there should be enough space in the bioreactor for it. That is one. And then you also will also have to set, say, the time. If you are having, say, maybe some 10 microliters of medium flowing at a second, then you have to calculate over, say, a period of, say, 5 hours, how much of medium should you prepare? How much of medium are you going to, make available to be pumped over this period? You shouldn't have a case where, especially when maybe a production is supposed to occur overnight, and at a point the medium finishes, and the pump will just be on and nothing is going to be flowing to it. So all of these things are considered, and it requires such mathematical calculations in order to be able to set up a production process. So these are very practical things that when you are in the industry, you will always be doing. You have a continuous culture. Also, if you're looking at the kinetics of the continuous culture, looking at that first equation that we looked at, which is the Monod equation, the steady state, and what we even refer to as washout, these states can also be represented mathematically. So what do we mean by the steady state? That you already understand, which is the stationary phase, where you have growth rate of the microorganisms, being this time around referred to in terms of the dilution rate. What we just looked at, the dilution rate being the ratio of the flow rate to the volume of the volume of the culture. And you see that that has a certain constant in there. You have the growth rates. In this case, if you have a steady state, what it means is that the growth rate is equal to dilution rate. The value which we just calculated from the previous slide. In a steady state, growth rate is equal to that dilution rate. And it also means that biomass concentration at that stage is expressed just with that Monod equation, where you have the maximum growth rates, the product of the maximum growth rate to the substrate concentration over the half saturation constant plus the substrate. Just like the Michaelis-Menten constant, you do have these representations of the steady state. And what do you mean by washout? What does it mean when we say washout? Now, if the dilution rate that you calculate, if that dilution rate exceeds the maximum specific rate, that is the Umax. Practically, what it means is that the amount of medium which is being pumped into the system over time, the rate at which it is being pumped is higher than the rate at which the microorganisms are growing. So what it means is that the microorganisms will not grow to their maximum, and you have a flow which is just washing these microorganisms out of the system. So they never reach their maximum state of growth and to produce the product, and they are just, being washed out of the system. That's why we call it washout. And it also means that there isn't going to be production of the product you are looking for, because you are just, is sort of just washing the system out, and everything is just moving in and going out, in, going out. The removal rate is higher than the growth rate. And so there will not be any production per se. And so at the end, you have what is being referred to as zero biomass concentration. Nothing is stored in, nothing is produced in there, and nothing can be harvested in the end. And if you look at it mathematically, looking at, say, substrate concentration being equal to, say, the product of half saturation, the product of half saturation constants with dilution rate divided by. So if you look at the denominator, which is a U max minus, that's growth rate, what it tells you is that if you're having the rate of the specific growth rate, which is not a specific growth rate, the dilution rate, which is D, if you are having it being more than the growth, the maximum growth rate, then it means this denominator is going to be negative. And negative means, you're not having any substrate. You can't have a negative substrate. Negative substrate means there's no substrate that is being accumulated. So that's what is meant by the zero biomass concentration, is to say that no product is being formed in this process, because you are just having a flow. It's just like you are just flowing some water through the system, and it will come in, move out, come in, move out. There's no time for the microbes to grow. So that kinetic curve, or the growth curve, which we saw for the microbes, will not be occurring. There will not be, say, a lag phase, exponential phase, stationary phase, dead phase. All that is not going to occur because there's no time for it. The medium just flows in, flows out, just getting flashed out like that. And so you have a negative substrate concentration, because you're now having that denominator, you have a dilution rate exceeding the maximum growth rate. And so if you do that simple calculation with the denominator, you find it's negative. Nothing is produced at the end at all. Now, so I think as we talk about the things, we did even speak about much of this significance of microbial growth kinetics already. Speaking of the fact that you really need to consider it in optimization of your system, the bioprocess optimization. Then, of course, your understanding of the metabolic activities of the microorganisms will give you lots of insight into how to engineer your system. Even if it's not at an industrial scale, maybe in the lab that you are doing some of this research, maybe some industrial research to understand how you can set up some of these systems. Because in any case, before some of these systems are set up, you do some industrial research in the lab, and then you scale it up to like some prototypes, small forms, and do some trials with some of these prototypes. If you see the viability of it, then you begin to say, okay, we can scale it up and have a bigger system that can produce at a large scale. At a large scale. Of course, you want to be able to determine if it's particularly a batch system, when the bioprocessing should stop and for harvesting to take time to take place. That is to determine the best harvest time, but that will be mostly just at the end of the stationary phase, just when you have the dead phase starting. At that point, you should be harvesting so that you don't have the system getting contaminated with a lot of dead cells and uh, toxic substances like the metabolites that are going to be getting produced in the dead phase. If a system is being designed specifically for treatment of wastewater, for instance, or for soil bioremediation, here too, yes, it's important for the design to consider microbial growth kinetics. I mean, if you're treating wastewater and at a particular time, when you have, the growth rates of the microbes not at the say maximum, for instance, for the best treatment of the wastewater talk. In the end, you think you have treated the water, but the water is not well treated, and especially in systems where you have wastewater treated and will have to come back into the mainstream, you can sort of cause intoxication of a whole population if the water is not well treated and it goes back into the mainstream system. And so these things have to be taken very, very seriously. Soil bioremediation, same thing. If a system for soil bioremediation is not effectively done, if it is not optimized, and you still have some significant amount of some toxic substances remaining in the soil, that can pose a lot of health threats also to populations. You have, even in the pharmaceutical industry, again, very, very important for the pharmaceutical industry, where microbes are being used to produce some active ingredients, maybe like production of things like insulin, even some production of some proteins, like some enzymes, like enzyme-based products. You do have optimization of the microbes that are being used in the fermenters or in the bioreactors, so that if the microbial growth kinetics is not very well considered there, that can pose lots of problems. So you can imagine the little thing which you learned, HND, BTech with microbiology with this growth kinetics is so much important in these different fields, waste management, pharmaceutical industry, food industry, all of it. You still will refer to this basic thing, which maybe when you were studying it, you just thought of it as one of those things, simple, and you study and that's it. But it's so much important you keep referring to it again and again, even at such industrial level. Yes, you see, today we're talking much more of like these quantitative values, talking about these equations that are being applied in the engineering of these bioreactors. You see that this quantitative work, that quantitative framework of representing these things with numbers is also having such a significance, such that we are able to control microbial growth under various conditions. Because I mean, you also remember that we did refer to factors affecting these things. I think we'll look at it again, these factors that affect microbial growth and enzymatic actions and things like that. Representing them with numbers makes it easier. It's not just some qualitative data which we are collecting, but it's quantitative. We are able to calculate and then be able to optimize the systems to have the best of results. Now we move on to kinetics of product formation. So what we did look at was kinetics of microbial growth. Now we move into kinetics of product formation. So our focus is now on, say, the product that is being produced. Now, the product that is being produced, you can look at it in two ways. That this product is growth associated or it is non-growth associated. So what do these mean? You have like a production rate of products which can be directly proportional to the growth rate of the microorganism, which means that you see the growth rate, which is that the exponential stage of growth of the microorganism. If you look at that curve and compare it to the formation of product, you see that there's a proportionality, which is to say that when growth rate increases, the product formation also increases. So if you see that, then it means the product formation is directly proportional to the growth rate. Now, you find products that are growth associated, being some primary metabolites of the microorganisms you use in the bioreactors. And these primary metabolites include amino acids, nucleotides, some fermentation products like ethanol, lactic acid. All of these are primary metabolites that are produced by the microorganisms. I mean, these are like, for instance, if you say amino acids, nucleotides, really, I mean, for instance, if you are using the microbes to produce some enzymes, you want to have some product. We did see the use of enzymes in different industries, like the food industry. How do you get those? Enzymes? You do produce them, some of them, by fermentation processes where you have microorganisms producing these things in bioreactors, and there you have proteins, which are the enzymes. Of course, these proteins are made up of these amino acids. If you have in production of some nucleic acids, for instance, of course, that's what you are referring to as nucleotides. Or in the process of fermentation, like we saw with the brewery industry, or maybe even biofuel industry, where we have a production of bioethanol, or maybe food industry in the production of yogurt, for instance. It's a production of lactic acid there. In those industries, those products which are being produced are primary metabolites, and so you have the product which is being formed being proportional to the growth rate of these microbes that are being used in the process. Now, because these primary metabolites are, you have the growth, the growth of the microbes and also the reproduction of the cells are directly proportional to the concentration of these primary metabolites that are being produced as the product of interest to us. Now, that growth associated, production is also something which it can be expressed mathematically. Because of course, if you are able to express something mathematically, you have the ability of controlling it more. And I mean, simply, you have the mathematical representation of this growth rate, dp dt, is equal to this constant, the growth associated product formation coefficient, and you have dx dt, which is the rate of biomass formation. You can say that is you can look at it as the rate of maybe consumption of the medium, for instance. You can look at it that way as well. And of course, in an integrated form, that is to simplify it, you say product concentration is equal to alpha x, where the alpha x is the growth associated product formation coefficient, or what we also refer to as yield coefficient times biomass concentration. What I said you can also refer to as, say, maybe biomass consumption, which is to say that the medium which is being introduced into the system, how much of it is being consumed. So you have, because of course the amount of microbes in the system will determine how much of the medium which is being consumed as well. And so you have that, simply, I think you prefer to make use of this integrated form, which is a much simpler one. But of course, maybe if you have, maybe you are given time, like say different times, beginning to the end, then of course you can calculate, say dt, and you are given, say, how much of a product is produced at a particular time. When you know there's no product at time zero, then of course you'll be able to have this, dp dt. Same way, if you are given this rate of biomass, the concentration of biomass, then of course with the time given, you'll be able to do those calculations in the industry and be able to set up your system. Now, so we move on to the next, which is the non-growth associated. If you say we have a product formation, which is growth associated, and we have the second one, which is non-growth associated. What is that non-growth associated? What it means is that the rate of product formation is not directly proportional to the growth rate of the microorganism, which means that exponential stage where you see with the microbial growth rates, if you compare it to the product formation, you don't see any correlation between them at all. So that is product which is formed not having any association with the growth rate. And this is usually for products that are formed after the growth phase, which means that the products are going to be formed at the stationary phase, when the microbes have already grown, and at that steady state, that is when the products are being formed, which is why you don't have any correlation with the exponential stage. And that's, these non-growth associated products include, substances that are produced by these microorganisms primarily for defending themselves. It's for defense mechanisms, and also for maybe signaling like say cell signaling. So if you do remember, I don't know if in your biochemistry you did things with hormones, some signaling molecules that like say the neurotransmitters. It is only with, say, already mature cells that will be producing these things to have such communication or to have such a protection for themselves. And so you see that these products are not produced by cells that are in their growing stages. They only get produced when they are already fully mature. And these are referred to as secondary metabolites. These are products like the antibiotics, which can be produced from microbes. We do have some of these microbes also producing things like some terpenoids, quinolines. These are some antioxidants which are produced by cells, already mature cells. Some of it can be some, maybe if it's some plant cells and things like that, they produce some glycosides. These are like complexes of some monosaccharides, with some modifications to them, with some phenolic compounds. Oxidation of these can form some of these quinolines. Some growth factors, pigments. You have these things being secondary metabolites of microbes, which are being used in bioreactors, and these are non-growth associated products. And so, like I said from the beginning, these substances have, they are used by these mature cells for defense mechanisms and for signaling. So, which is why you find them mostly in already mature cells. And the non-growth associated product formation can also be represented mathematically, just like we did with the growth associated. We have a constant beta, which is the non-growth associated products formation coefficient, which we also refer to as a yield coefficient. Same thing with biomass concentration, which is X, dPdt, the rate of product formation. In an integrated form, you have the product concentration being equal to the constant, the yield constant, times the biomass concentration, and time, the time over which you have the production occurring. But these are formulas which I think we'll look at it more pictorially so you can have a better understanding of it. You have, apart from, you see these two, which is the growth associated product formation, and then a non-growth associated product formation. It is possible to have a mixture of these two occurring in a production process. And that is referred to as a mixed growth associated product formation. So you have the products that are being formed, some of it being formed in the exponential stage, the growth stage of the microbes. And you have some of the products that are formed at the stationary phase. So you have this mixture of primary and secondary metabolites. You do have these things occurring, which will mean that the rate of product formation will have components that are proportional to the growth rate, and you have components that are independent of the growth rate. So you have a certain as if you are having a certain correlation or say proportionality with the growth rates, not fully, but then you also have some slight proportionality also with the stationary phase. You have such a mixed growth associated product formation also in bioprocessing systems. And this mathematically is also represented by what we refer to as the Lütken-Parrot equation, which is a summation of the growth associated system with the non-growth associated system. And so you can see clearly that the rate of product formation is the sum of the rate of product formation in the growth associated and the rate of product formation in the non-growth associated system. Clearly, this can also be written in the integrated form, much simpler form, where you have the products, the product concentration being equal to, say, the alpha, which is the constants, the growth associated constant, growth associated yield constant, the product of that with the difference in concentrations of the say initial biomass and the biomass concentration at a particular time that this is being determined, and you add that to the product formation occurring with the non-growth associated, where the constant there is beta, which is the non-growth associated yield coefficient, the x being the biomass concentration, and t is time. So this has a summation of what is happening with growth associated system and then the non-growth associated system, summation of that, which is being referred to as the Liudkin-Parrott equation, to represent that mixed growth associated product formation. Right. Now, yes, so graphically we can see this where you have the growth associated. Where we see X, referring to say the product. Actually, I think P, the colors are not matching. So let's see, P, which is blue, should actually be the yellow, which is the product which is being formed, and the X, you can look at it as the growth rate. So you can see that for growth associated curve, you see that product formation and the growth rates are proportional. You can see this clearly, are proportional. Then you have the mixed growth rate. You see, we saw that in the mixed growth rate, you have some components of proportionality with the exponential phase, that is the growth phase. At the same time, you have some components with a stationary phase as well. So you see that there isn't that exact proportionality, but you can see that there is that a mixture of these proportionalities. Again, here the P, which is the product which is being formed, is correlating with the yellow. So we could have done this yellow rather blue, so that it will match with the color of the P, which is a product, and then the X being the growth rate, which is the red there. Then you have non-growth associated. The non-growth associated. So here clearly you see that there's no correlation at all. The product formation is independent of the growth rate. The growth rate, which is X, is clearly independent of it. As looking at it with a graphical representation in this way, I think you have a better understanding of what we mean by those things with those formulas and explanations which have been given so far for these types of product formation kinetics. Right. Now, we have, this is to do some comparisons with product formation in different culture systems, because so far we have, from last lecture, we've looked at the batch system, the fed batch system, the continuous system. And so if you compare these systems in terms of product formation, what are some of the differences? Major differences that you find out if you take into consideration different aspects of these systems. So the first is like the system itself, the type of the system itself. You realize a batch system is a closed system because you add everything in there, you close it and leave it till the end. The fed batch system we saw is semi-closed because it is only at some specific intervals that you feed in the medium, and then the process runs to the end till harvest time. Continuous system, you have the flow of the medium consistently throughout the process. So it's an open system. That's an open system. If you look at the phases where you have like production of the product, like where the product is being formed, in a batch system, obviously the product is going to be formed at all the stages. I mean, that is of course not at a lagging phase per se. You have a product formed at the exponential stage, and also at the stationary phase, because you are waiting for the whole process to finish over a certain period of time before you do harvest. So you have a production of these products, in these phases. How about fed batch? I mean, what does a fed batch system do? The fed batch system uh, extends the exponential phase, so because it keeps introducing medium into the reactor, and that extends the exponential phase. Like more and more of the cells are going to be produced. There's like reproduction increases, and once reproduction of the cells increases, then you have the exponential phase, the growth of the cells, that period also extending. And at the same time, that affects also the stationary phase, because once you have more cells that are being produced in that exponential phase, then of course the stationary phase is also going to extend. And so you have some of the products produced in the exponential phase and some of it in the stationary phase. In a continuous system, you have some of the product also, just like the fed batch system, being produced in the exponential state, some of it in the steady state. But you have in most part of it, primarily the production in a continuous state, continuous system occurring in a steady state, because the system is controlled in such a way that you have production which is occurring with already mature cells, so that even though you have a flow of medium into the system constantly, it gives the opportunity for more cells to be reproduced, grow, and reach the steady state. And in the steady state, you have the production of these products. So mostly, the continuous system is used when it's known that a product is formed at a stationary phase, so that with such a continuous system, you increase or you extend more the stationary phase, so that you can have the best of yield. If not, I mean, that stationary phase will be so short that the amount of product which is going to be produced will not be that much enough, and economically that is not going to be viable. And so you have, I mean, if you look at the things which we have just looked at, the things which we have just learned, and you apply it, then of course in a batch system you have growth, non-growth, mixed, product formation. I mean, because we said the products which are being formed are formed in all the phases. So which means you have a mixture. You have some of the products which are in the growth phase. You have some which are in the stationary phase. So if you put all that together, then of course you can classify that batch system to be growth, non-growth, and mixed associated growth systems. Fed batch, similar, the same thing, just like the batch system is also having a mixture of everything. If you come to the continuous system, because you have production mainly in a primary in the steady state, it will not be related so much to the growth stage. But here again, because you keep introducing more and more of the medium, the more of the medium you introduce, the more cells are growing, and the more the stationary phase is extended. So here too, you can have some products that are related to, say, the mixed product formation system, and some of it with a growth stage rather than the stationary phase. You do have the control of nutrients. You see, the nutrients are where the nutrients are, in the medium that you are introducing. And you see, if you compare the system based on that, then of course in a batch system you have a fixed initial amount which is introduced in the system, closed. Nothing else is added to it. So you have just the fixed initial amount, and that's it, nothing else. Fed batch will have a controlled addition. At specific times, you do add some amount of medium again, and so you are adding more nutrient there in the system. Continuous, you are having continuous supply from the beginning all the way to the end, and so you have a continuous supply. How about productivity? You can see clearly, I mean, the last time we did see that actually each of these, like if you take a fed batch, the fed batch system is an improvement of the batch system, whilst the continuous system is a further improvement to increase yield. And so productivity for the continuous is very high, whilst you have fed batch, I mean high, and you have the batch system moderate. But of course, each of them are used for different purposes. Complexity, the batch system is the simplest because you just put everything there, you close it, you wait till the end. Fed batch is moderately complex, and a continuous system is more a complex system. We'll see, we'll have some virtual excursions so that you can see really the systems in action, how particularly this continuous system, which is the most used, I mean, in modern times, you'll see it being used. So you can see how complex such a system is with, the pumps, the bioreactor, the computer systems which are connected to control all the conditions and things like that. Contamination risk. So you see, even though we say that the continuous system is having a high yield, you see that the continuous system at the same time is having a high risk of contamination. The fed batch moderates, and the batch system is having a low risk of contamination. Why? Because you just put everything together, you close it, and once it's not contaminated from the beginning and is close to the end, then the risk of contamination is reduced. But with a continuous system where you have a continuous flow of medium into the system, there is that high risk of some contamination along the line. Fed batch, because the feeding in of medium along the line is controlled, and so you have the risk here too being moderate. And then these are some examples of products that are formed that these systems are used for. So for instance, ethanol, for instance, you have a batch system being used, antibiotics, a batch system being used. So in the pharmaceutical industry, for instance, you use this batch system, everything is fed in, the bioreactor is closed, and then the time is set, the wait till the end, and then you have the products which can then be harvested. You have fed batch system being used for, like, say, biofuels. You do remember that we have the breakdown of various carbohydrates, different ones with different enzymes, and sometimes some of these breakdown of some specific carbohydrates have to happen at specific times. So if you're having some pectins, you're having some xylan, you have different carbohydrates with different types of bonds, being the ones having a branch chain like cellulose, dextrins, all of them with different enzymes that are being introduced, like amylases, cellulases, xylanase. All of these will have to, say, pectinase. All of these will have to break down these specific carbohydrates in order for the best of yield to occur. not only for biofuels, but even for maybe production of, say, in the breweries, beer, for instance. You have the biomass for a product. You have continuous enzyme production. That is a major example for the continuous system. The optimal use of this batch system is best for, say, for experimental purposes, for basic research in the lab. You set up a batch system, you close it, a reaction occurs, and then later you take products and you analyze. Industrial fermentation, so to produce high value products, the fed batch system is more economical there to use. And even now, the continuous system is the one with very high productivity, as we've seen earlier. For things like waste treatment, continuous systems are used. Because this has to run, imagine the amount of work which has to be done with waste management of, say, urban waste, for instance. It's a lot of work, which if you say you want to be doing batch or fed batch, I mean, there's going to be a lot of backlog. So you need such a consistent, continuous system to be able to have, the optimum productivity coming out of these industries. Now, if you do compare, say, environmental factors, you see, this time around, you are looking at, you see, the same thing we looked at with factors that affect enzyme activity. Have you seen? These same factors are coming here, temperature. And these are the factors which are controlled in the bioreactors. When the bioreactors are being set up, you do set up, say, temperature. There should be an optimal temperature range for different microorganisms. Because, of course, you are dealing with metabolic processes, and you're talking about enzyme activity. So you need to optimize temperature. pH, the same thing. We did look at it when we were looking at enzymology. So again, for enzyme activity, stability of the cell, you need to optimize pH as well. This time around, you are looking at it as environmental factors, because these are the conditions which you are optimizing in a bioreactor in such an industrial system. Then there is one other thing which is specific to this industrial process, which is oxygen availability. This is in terms of, say, the reaction which you are running. Is it aerobic or anaerobic? Which product are you producing? If you're producing ethanol, for instance, and then you have too much of oxygen that is entering to the system, obviously you are not going to produce ethanol. You are going to then have a reaction that will just be an aerobic breakdown of carbohydrates, which will just result into some acetyl-CoA, carbon dioxide, and that's it. You don't have ethanol. You need anaerobic condition. And so you need to control the amount of oxygen in the system. And then, of course, nutrient availability, you can look at it as what we saw in biochemistry as substrate concentration. But in this case, you see, you also have to bring your mind to something. There is something very interesting about biological systems, which is if you look at, the biological structures which we have looked at so far, carbohydrates, proteins, nucleic acids, lipids. If you look at all of them, you realize that they all have carbon skeleton. So carbohydrates, I mean, if you look at the monosaccharides, they all have carbon, 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 carbon. And then what next? You have hydrogen, hydrogen. Then you have oxygen in all those molecules. Then if you come to proteins, you will see in the amino acids, you have some nitrogen there. And then if you come to, say, the nucleic acids, you talk of phosphodiester bonds. You have phosphate groups now on these nucleotides. And then, of course, in some of these amino acids, you have sulfur, like methionine, cysteine, you have sulfur in them. So in fact, if you pay attention to these major building blocks of biological systems, you realize that the major elements that are found in biological systems are carbon, hydrogen, oxygen, nitrogen, sulfur, phosphorus. These are major elements that you find in biological systems. I mean, the rest, I mean, you do refer to, micro elements, for instance. But what I've just mentioned are like say major components that you find in all these biological systems, cells, the biomolecules that form, these cells. This is what you find in them significantly. And that's the reason why when you are growing microorganisms, say the medium that you use in your microbiology, if you go to look at these mediums, maybe those of you who are microbiology experts, I am not. Just take a look at the components of this medium, and you realize that it is providing carbon, hydrogen, oxygen, nitrogen, phosphorus. I mean, depending on the microorganism you are growing, you want to have a certain percentage of these elements in the medium. Even if you come to, for instance, agriculture, you see fertilizer, for instance. What are the elements which you have, being, major? You have nitrogen, you have phosphorus, you have potassium, because these elements are important for the growth of, plants. And you have different fertilizers for different purposes. And you see that they are enriched with specific elements like that. And these are like they are limiting, or say limiting nutrients that can shift metabolic pathways and affect the yield of the products in bioreactors. All right, so I mean application of those factors which we learned in enzymology now in industrial systems. So you see what I was talking about. You have these different sources for different elements, like you have carbon sources, which are say maybe glucose, that is sugar, lactose, some milk sugar, glycerol, and coming from lipids. These are some sources of the elements. Carbon uh, for the microbials, for the microbes to feed on as medium. And you have nitrogen sources, of course. Amino acids have nitrogen there. They are in the amine group. Or you have ammonia, nitrates will be good sources for nitrogen. You have phosphates, sulfates that will provide the sulfur and the phosphates. I refer to the element sulfur, the elements phosphorus, which are found in these biological, building blocks. So you see, each and every one of these nutrients have some, specific sources which are used in the production of the medium, which is introduced into the bioreactors, and they are used to achieve production of specific products. Yeah. So, for instance, if there is a need to produce substances which, let's say, some enzymes which are proteins, then of course you will need a lot of these amino acids. You need some nitrogen there, a lot of this nitrogen in there. How about if it is nucleic acids? If it's nucleic acid, of course, you need to have phosphate in there to be able to have a good yield of nucleic acids produced, because the nucleotides have phosphate group in them. So you require phosphorus there, for a good yield of the nucleotides. Other factors that are also manipulated in these processes are, the gene expression. We did see gene expression to be in protein synthesis, where you can have a regulation of genes in order to have the production of particular products depending on the genes that are expressed, because the expression of it will produce specific products, proteins that are catalyzed, the protein synthesis, that are coming from some specific genes. You have also regulation of these metabolic processes, like the gene expression, with some processes of, say, repression or regulation of just maybe, say, nitrogen, a particular substrate, will result into regulation of a particular production, metabolic process in the cells. Then you have some of this genetic engineering, modifying of cells, like maybe it's even some recombinant DNAs that are used also for regulation of these processes. This is just to say that, for instance, if you want to have a better yield of some products, you take a particular gene and have maybe some genetic some recombination of plasmid, introduce it into maybe yeast. So you see, naturally, the yeast is not to produce that particular protein, but you would have engineered it by such a recombinant DNA, so that the yeast begins to produce the protein that you need. So that is manipulation of this biological system. Of course, this then will become what you are referring to as a genetically modified product, things like that. I think this should be the last, which is also things we have done with, say, the metabolic pathways like glycolysis, the TCA cycle, the pentose phosphate pathway. All these pathways, the specific products that they produce, and knowing the pathways, the enzymes that act in these pathways, the conditions that are required in these pathways, then you have the knowledge to be able to control the system such that, so for instance, if you were doing, like I mentioned earlier, fermentation to produce ethanol, you don't want your system to go through like a TCA cycle. You want it to go into fermentation. And so you regulate it, controlling oxygen. So you create an anaerobic condition there. If you're talking about production of some secondary metabolites, like some antibiotics, pigments, there too you often will have to create some specific conditions, limit some nutrients. To create, it is some form of creating a stressful condition for the cells, the microbial cells, so that they begin to produce the antibiotic in response to it, because this is for defense. The substance that is being produced is for defense of the cell. So you have to create a stressful condition in order for the cell to respond to it, to produce such a product for you. We did see anabolic pathways, which we also refer to as biosynthesis. So the products that you want, are they, if they are not products that are formed in catabolic pathways, then of course there must be a condition created for such biosynthesis to occur. Biosynthesis will occur if the cell lacks a particular compound, and so we have to produce it. But if you have the substance, because we have seen regulation to see that even inside biosynthetic processes, you have the products acting as um, um, inhibitors, giving a feedback inhibition to these enzymes to inhibit them to stop the process and all of, and you have to control all of these things with some serious engineering optimization to have the flow of metabolites towards the target product. All right. So I think this is where I will say thank you for your attention, and then I will open the floor. If you have anything, any question, any contributions, you can write in the chat. I respond, or you can raise your hand. I will enable your microphone. You unmute yourself, and then you can speak. The floor is open.